Hello everyone, welcome back to the class. In our previous class we discussed about mitosis and its different substages, and we completed the cell cycle part, that is mitosis cell cycle part, by the end of cytokinesis in plant cell and cytokinesis in animal cell, and also we discussed about syncytium and cenocyte. Today we'll be discussing about meiosis, which is another type of cell division where the number of chromosomes is reduced to half. Meiosis is the term coined by Farmer and Murray. It is a specialized form of cell division in which number of chromosomes is reduced to half. The reduction in chromosome number is achieved by one round of DNA replication being followed by 2 rounds of chromosome segregation with no intervening round of DNA replication. So this will be understood in further classes. So meiosis is divided into two parts, that is meiosis 1 and meiosis 2. At the end of meiotic process there are 4 daughter cells rather than the 2 produced at the end of the mitotic cell division. Each of the resulting daughter cells have 1/2 of the number of chromosomes as the parent cell. So these are the events of meiosis. 2 pairs of homologous chromosomes will be present, and there is a predominant centromere will be present. Pre-meiotic in S phase, it undergoes pre-meiotic S phase. Then there will be formation of paired sister centromeres. Meiotic prophase, it undergoes meiotic prophase, where meiotic prophase is the major entry into meiosis. Meiotic prophase is divided into 5 substages, that is leptotin, zygotine, pectine, diplotin and diakinesis, based on the nature of the nature of the chromosome or the chromatid strands. Then after that meiosis 1 will continue by metaphase 1, anaphase 1 and telophase 1. Then there is a gap between the meiosis 1 and 2. That gap is known as interphase stage. After that meiosis 2 will be continuing, that is prophase 2, metaphase 2, anaphase 2 and telophase 2, resulting in haploid cells, haploid 4 daughter cells. Meiosis 1 is subdivided into prophase 1, anaphase 1, prophase 1, metaphase 1, anaphase 1 and telophase 1. Further, this prophase 1 of meiosis 1 is subdivided into 5 stages: leptotin, zygotine, pectine, diplotin and diakinesis. So let us know about these substages in today's class. This leptotin, although each chromosome has replicated and consists of 2 sister chromatids, so these chromatids are unusually closely opposed, and each chromosome therefore appears to be a single separate chromatid, will not become visible until later in prophase at either the diplotin stage or in diakinesis stage. So what happens in this stage is that the chromosome ends are attached to the inner nuclear envelope. In the leptotin to zygotine transition, the tips of these chromosomes move until most end up in the limited region near others. So this form of arrangement is called bouquet stage. Leptotin in this word, the word lepto means thin. So it represents that the chromatids are thin and they form a bouquet shape. So therefore this form of arrangement is called as bouquet stage. Bouquet formation directly facilitates homologous chromosome pairing and synapses. See here in this leptotin stage, so this is the cell, so the chromosome ends are attached to the nuclear envelope to the nuclear envelope, the chromosome ends. This later chromosome ends the tips of the chromosome move until most end up in the limited region. So this form of arrangement is called bouquet stage. Here the chromatids are visible, but they are thin, and you can see centromere also. Followed by this leptotene, there is next stage, that is zygotene. So it begins as soon as the synapses or intimate pairing between the two homologs is initiated. The word synapses here refers to intimate pairing between the two homologs which is initiated in this stage. Consider there are two parents here, that is, this is male parent and this is female parent. They both contributing a homologous chromosome, or I'll write it in this. Okay, so both are contributing. I'll better write in this different color. So this is male parent's chromosome and this is female parent's chromosome. Okay, so these are homologous in structure. Homologous in the sense wherever the genes are present, they resemble each other. So this is homologous. The structure and the inner content of the chromosome should be equal. So these are homologous chromosomes. Okay, they come together here. It forms a synapse. It forms a intimate pairing, or I'll write it like this. It forms an intimate pairing. Okay, so this is the maternal and paternal copy of each chromosome recognizing each other. Okay, this synapses often starts when the homologous ends of the two chromosomes are brought together. So these are brought together on the nuclear envelope and they proceed inward in a zipper-like manner. The zipper, to be precise, here there is a zipper. It will intimately pair inward in a zipper-like manner from both ends. So in other cases, this synapses may begin in internal regions of the chromosomes and proceed towards the end. Okay, so often it is inward, from outer to inward. So what happens in sex chromosomes and pairing of sex chromosomes? So these are chromosomes which are resembling similar in the size and shape, but when it comes to sex chromosomes, X chromosome, female chromosome is like this, and Y chromosome, male chromosome, is somewhat short. So what happens in this case? So the pairing of sex chromosomes occurs in similar fashion as homologous autosomes. So mammalian females have two X chromosomes, right? So they have two X chromosomes, and the paternal mammalian males have one X and one Y chromosomes. Although these chromosomes are not homologous completely, so they too must pair and undergo crossing over, right? So pairing and crossing over are possible because of small region of homology. So there are some small regions of homology which makes them to pair. So the two chromosomes pair and cross over in this region, ensuring that each sperm receives either Y chromosome or X chromosome. And now each pair of homologous chromosomes is undergoing intimate pairing. as written already. So it is undergoing ultimate and intimate pairing. So I'll write from outward to inward. So now this results in bivalent. This results in bivalent, or it sometimes is called tetrad. Tetrad means that there are 4 arms. So therefore it is tetrad. It is bivalent. Now it has become 2. So it is known as bivalent. So there is one complex which helps in the synapses process. So that is synaptonemal complex, which mediate chromosome pairing. The synaptonemal complex will be discussed in next class. Thank you.